Welcome to the nSolver 2 webinar series. In this webinar series, we're covering the enhancements that have been added to nSolver 2. And in this particular webinar, we're going to concentrate on the basic visualization and the user-defined gene annotations. I'd just like to point out that the heat maps we'll cover in a separate webinar. While the primary focus of this webinar is visualizations, all of the visualizations are much more robust when your genes have been annotated. Custom annotations are informative generally, but particularly useful for filtering when creating visualizations. To enter annotations to genes, navigate to the Raw Data tab and select the RLF file containing the genes you want to annotate. Then select the genes table. I have a spreadsheet here of annotated genes, and I can simply paste this into my list of annotations once I've made sure that my filtered list of genes is sorted identically to my spreadsheet. No saving is needed, and this can be done at any time before or after creating a document. It's important to remember that this field is a text field. If it is being used for ordering purposes within the analysis wizard, then it may be advisable to use letters A to Z in the beginning of this field so it sorts properly, as numbers are sorted as text fields. For example, 20 will be sorted prior to 3. These genes can be visualized and exported in all subsequent nSolver tables and visualizations for this code set. We've now in version 2 added an additional icon for analysis. Analysis can be run from any of the raw, normalized, grouped and ratio data levels. The analysis wizard can be launched after selecting the desired data. On the first screen of the analysis wizard, you enter the name of the analysis and the analysis type. You can also perform various high-level filtering. nSolver 2 provides heat maps, scatter plots, box plots, violin plots and histograms. In this webinar, I'm going to cover all of these analyses with the exception of the heat map, because due to the large number of choices available, we're going to cover that in a separate webinar. I'm going to begin by creating a scatter plot for my group data. It's important to note though that all the screens are the same for each option, so I'll only demonstrate the initial parts of the analysis wizard once. The filters you can choose are to filter your outlier samples and genes from the analysis using the histogram. The histogram filter is useful to assist in filtering outliers from the analysis, particularly valuable for large sample sets. Uncheck this option if you want to completely bypass the histogram filter page. You can exclude samples by QC or normalization flags, and checking this option will exclude any samples that had QC or normalization flags and can save you from excluding them later on if that's your intention. You can exclude all controls, and this will exclude all probes that have a code class of positive, negative, spike in or invariant. And you can exclude all content normalization probes. This will exclude all probes used for content normalization, that is your reference or housekeeping genes. Once you've selected the filters, you can then proceed through the wizard. If you selected the checkbox to filter samples and genes using the histogram filter, the histogram filter is the next page of the wizard. Otherwise, the histogram filter page will be skipped and the next page you see is the exclude samples page. The histogram filter is generally designed to allow for removal of poor quality samples or genes from downstream analysis and visualizations. At the top, the histogram can be adjusted to appear more or less granular by adjusting the number of bins or the bin size. The graph on the right is for gene filtering and displays log2 counts on the x-axis and number of genes on the y-axis. You can use the following functions to remove outliers. You can move the slider bars to the right or left to select which genes per sample to be included. As you do this, the number of genes included and excluded will be dynamically updated at the bottom. You can edit the bounds manually. For genes that are outside of the bounds specified, samples are excluded for further analysis only if the specified percentage of the sample is outside the upper and lower bounds. The graph on the left is the same principle, but is for filtering samples and will remove samples with less than the specified percentage of genes that are outside of the interval specified by the upper and lower bounds. So after specifying any desired sample and filtering options, the samples and genes specified by the histogram filter are carried over to the next 2 pages of the wizard and are classified as either included or excluded from the analysis or visualization. The next page of the analysis wizards allows for viewing of samples that are set to be included and excluded from the analysis. Any samples that are excluded based on filtering from the first 2 screens are displayed here and can be adjusted. Sample list can be adjusted manually by selecting the samples and using the right and left arrows. The genome probes to be included and excluded from the analysis are shown on this page of the analysis window. Similar to the previous page, you can adjust these probes manually by selecting the probes and using the right or left arrows. Selecting Next will now bring me to the scatter plot specific window. For scatter plots, I have to include at least 2 samples to include in the scatter plot. In this example, I'm going to choose all 6. The scatter plot is generally a visualization that plots one of the samples on the x-axis and the remaining samples on the y-axis. I can always adjust the x-axis sample from the drop-down menu. So in this case, let's compare to normal and display all of the other. I can dynamically adjust the plot to add or remove samples by selecting the checkboxes on the y-axis. So in this case, I'm just comparing the normal to the loom A and loom B. For box plots, violin plots and scatter plots, there are some common features I'm next going to show that allow for plots to be created in a publication-ready format. The file menu is available from the menu bar at the top. You can perform the following using the file bar. You can save the image as either a JPEG, PNG or GIF format. You can save the settings. The Save Settings preserves the custom adjustments you've made to titles, fonts, and the overall look and feel of the visualization. And you can print to your designated printer. In the Settings menu, there are a number of common features to all analysis. You can set the scale type to linear, logarithmic 2 or log base 10. I can change the legend and data points. So I can choose to display a legend or not. And I can also change data points so that they are easier to view. So for instance, I can change the shape and I can also change the color. One feature in common settings that's unique to the scatter plot only is the functionality to perform a linear regression. I can select the linear regression and either show the R-squared, the equation, or both. And they'll then appear on my chart. It's important to notice I can move the legend box around so that this is more clearly visible. And one final functionality in the scatter plot is the ability to hover the mouse over an individual data point and see which gene is actually being represented by that data point. Once you've finished with your plot, printed it, if needed, save the settings, then close. We'll close that analysis. But it's important to notice the analysis is stored here in the analysis data section. And we can review it by going back to analysis data. Violin plots are a convenient way of graphically describing subsets of your experiment with a curve around the plot to show the rounded distribution of data. Selecting genes of interest from the left-hand side dynamically builds the plot on the right-hand side, as you can see here. Violin plots have blue lines extending vertically, indicating the lower adjusted value, first quartile, third quartile, and upper adjusted value. The lower adjusted value and upper adjusted value are the minimum and maximum values in the data series. The median is depicted with a red dot. The violin plot window contains several features you can use to quickly identify the genes you want to plot. The first is the filter function that's available throughout nSolver. But here let's, for instance, look for all of the genes that I've annotated as driver genes in this set. And now only the driver genes are available. So it makes it much easier for me to sort through them all and look at the results. nSolver also provides you the capability to display the violin plot by condition. If experiment annotation columns were added to create treatment groups of data, this option allows for a useful visual of a gene or small group of genes plotted across the various experimental annotations. So if we select Show by Condition in this example, we see all the subtypes and the different expression of that gene. When this becomes really powerful is when you have 2 sets of annotations. So let me just turn to a smaller, different set of data. Small set of data here, I've actually got multiple treatments, A, B and C, and multiple time points for each of those treatments. In this case, if we select a gene and show by condition, we get the chance to see both the treatment and the time. In this data set where we only have 3 data points, the violin plot obviously does not show a lot of detail, but we can see the median points differ with the different time points. And we can also view just the treatment group or just the time point in this experiment. A very powerful tool for analysing your data. Box plots are a convenient way of graphically depicting subsets of your experiment through their quartiles. Box plots have lines extending vertically from their boxes, the so-called whiskers, indicating variability outside the upper and lower quartiles. Outliers may be plotted as individual points. Box plots display differences between subsets of an experiment without making any assumptions about the underlying statistical distribution. They're non-parametric. Box plots in nSolver function similarly to violin plots. You select the genes of interest from the left-hand side to dynamically build the plot on the right-hand side. Again, there are several features you can use to quickly identify the genes you want to plot. The first is the filter function, as available throughout nSolver. And again, you can display the box plot by condition. And as I showed previously, the settings menu and file menu allow you to set plot settings, specify chart tables, save options and print. The histogram plot represents the count values of all genes across all samples included in the analysis. The height of each bar represents the frequency of each count defined by the bins. Similar to all visualizations in nSolver, this plot is also available at all data levels. You can display a more granular view by adjusting the number of bins or the bin size. So here we are seeing how many genes have a certain number of counts. This is in log 2. All analyses are saved automatically upon completion of the wizard. Analyses are saved per experiment and can be accessed via the experiment tree from the analysis data level. To view a previously created analysis, select the desired analysis and click the analysis data icon as. The name and description of an analysis can be edited in the main view of the data. Simply click in the analysis field or description and type in the new text field. These fields will automatically be saved. As a hint, within nSolver, when the column title is bold, it means it's editable. Analysis can also be deleted. Just select the analysis and delete. You'll be asked for confirmation. Finally, an analysis report is available in a similar format and content to the experiment report. This report contains all of the information about the data used for analysis, as well as the parameters used in the analysis. Thank you very much for listening to this webinar. And remember, all the details we've talked about are in the nSolver user manual, which was included with your installation files. And if you have any problems, please contact nSolver support@nanostring.com. And of course, as always, any general nCounter questions, contact support@nanostring.com. And for any sales, contact sales@nanostring.com. I hope this webinar was useful, and I look forward to meeting you in the next webinar.